Antiviral therapy of COVID-19.
Abstract. Since the beginning of the COVID-19 pandemic, the scientific community has focused on prophylactic vaccine development. In parallel, the appearance of the pharmacotherapy of this disease had increased due to the declining protective capacity of vaccine agents, new strains, as well as increased knowledge about the structure and biology of the pathogen. Control of the disease has shifted to the focus of antiviral drug development over the past year. Introduction. The SARS-CoV-2 infection or COVID-19 pandemic continued to pose a threat to public health. The number of registered cases of infection exceeded 641 million and more than 6.8 million infected people died. About 270,000 cases of infection are registered daily. The main direction of COVID-19 contraction or vaccination, prevention of the development of the disease, and treatment of infected patients. Knowledge of SARS-CoV-2 routes of transmission, its molecular organization, life cycle, future and cellular infection mechanism allowed us to improve treatment efficacy using various types of drugs. The accumulation of practical experience and the results of scientific research made it possible to improve therapeutic strategy and provided the basis for the development of new types of antiviral drugs, specifically blocking viral replication by suppressing the activity of the corresponding site of the virus genome. Through the life cycle of SARS-CoV-2, the coronavirus is a single-strand RNA virus with a positive sense genome. This undergoes significant mutation. It is divided into 4 generations based on infection: alpha coronavirus, beta coronavirus, delta coronavirus, and gamma coronavirus. Coronavirus mostly cause respiratory and gastrointestinal disease. Common symptoms include fever, cough, fatigue, body aches, weakness, and mucus secretion.
2.1 Attachment to target cell and fusion. The main target cell of SARS-CoV-2 in epithelial cell lining the respiratory tract, lung cell, and alveoli. The initial step of infusion involves attachment to target cell, can fusion with the S protein of SARS-CoV-2, binding to enzyme produced by the host, such as transmembrane protein X, serine 2.3 Replication of viral RNA and mRNA sentences. The replication process of RNA virus is carried unbind the RTC complex, with the main player being the protein NSP12 RNA. The dependent RNA polymerase. This replication results in the production of full-length genome which serves as template for censored RNA. The generally RNA is that usually for transferring non-sexual viral protein and also contributes to the assembly of viral replicative complex. 2.4 Translation Sexual protein S, E, M, and N and sexually protein and translates for the receptive mRNA structure. The sexual protein are essential for assembling mature viral particles that grow, shower, accessory protein play non-role in virus replication.
2.5 Viral assembly and budding structural protein SEM and N of SARS-CoV-2 are synthesis in the endoplasmic reticulum and released extracellularly through vesicle mediated processing. Assembly involve remodeling of cellular structures, endomembrane, and proteasome, creating RNA synthesis fairly compartment and shielding viral RNA from degradation. This structure form an intercellular network connected to ER.
Ethiotropic therapy COVID-19 official recommendations and medications. The antiviral therapies for COVID-19 include immunoglobulins, monoclonal antibodies, nucleoside analogs, protease inhibitors, fusion inhibitors, and antisense nucleotides. These therapies target different aspects of the virus' life cycle to inhibit its replication or spread. Each therapy has a unique mechanism of action. The official status and recommendations for these drugs can vary, so it is essential to refer to reliable sources for the latest information. 3.1 Plasma of convalescent and preparations based on monoclonal antibodies. Treatment options for COVID-19 include using plasma from recovered individuals or monoclonal antibodies targeting the virus. While convalescent plasma show mixed results in studies, monoclonal antibodies like bamlanivimab and etesevimab are promising for mild to moderate cases. Other strategies involve reducing the inflammatory response, which drugs like tocilizumab and sarilumab, and studying tyrosine kinase inhibitors for their potential effectiveness and safety in COVID-19 patients. This is a picture of 3.1. Part 2 Interferons. Interferon IFNs are essential for the body's defense against viruses, but their effectiveness in treating COVID-19 remains uncertain. While some studies suggest potential benefits, others have found no significant improvement in patient outcomes. Type 3 IFNs show promise in reducing viral shedding, but more research is needed. Virus Viruses like SARS-CoV-2 have way to evade IFN effects, and prolonged IFN exposure can increase inflammation. Increase inflammation. Timing and duration of IFN therapy are factor, crucial factors. Currently, global guidelines do not recommend interferon for COVID-19 to treatment.
Next will be discuss of nucleoside analog inhibitor of replications. One, favipiravir. Favipiravir is a modified pyrazine analog that was initially used in influenza and Ebola. The drug inhibits RNA-dependent RNA polymerase (RDRP), which is necessary of viral genome, which divides the spread of SARS-CoV-2. Favipiravir was one for the first medication study for the treatment of COVID-19. Next, ribavirin. Ribavirin is a synthetic nucleoside analog of ribofuranols. Ribavirin incorporates in viral RNA and therapy inhibits its synthesis, causing mutation in the viral genome and suppressing normal replication of the virus. Next, remdesivir. Remdesivir was invented by Gilead Sciences to treat infections caused by Ebola and Marburg virus. Later, its ability to suppress the replication of the SARS-CoV-2 clinical isolate was demonstrated in vitro experiment in cell culture. The study of the drug is ongoing. 119 clinical trials of remdesivir are being conducted. Remdesivir administration should be recommended within a week of onset of symptoms and not recommended for patients with critical COVID-19. Next, molnupiravir. Merck and Richard Biotherapy have developed the drug molnupiravir, a broad-spectrum antiviral agent. Molnupiravir is able to introduce error into genome RNA of coronavirus by replication gene nucleotide which A and C which U. During replication, molnupiravir is currently recommended for use in the patient with non-severe COVID-19 and high risk of hospitalizations. And next, we will discuss of small molecule-based therapy. 1. Protease inhibitor. For many viruses, the protease enzyme play a critical role in viral protein maturation by cleaving translate polypeptide to functional product. As a result, virus proteases are often potential drug targets. For instance, HIV-1 protease inhibitor, tipiranavir, duranavir, and piravir are among the FDA-approved drugs. In the context of the pandemic, many researchers have turned their attention to existing viral enzyme inhibitors to evaluate their potential in COVID-19 therapy. Next, fusion inhibitor umifinovir. Umifinovir is an antiviral drug for the treatment of influenza. The drug inhibits the penetration of the virus into target cells and stimulates the immune response. A recent phase 3 clinical trial showed that treatment with umifinovir is effective, safe, and well tolerated in patients with mild asymptomatic COVID-19.
All of the above-mentioned drugs or similar drugs are being used to treat COVID-19 as a result of repurposing their primary biological effect.
This slide explains its limited effectiveness against SARS-CoV-2, deciphering the SARS-CoV-2 genome and studying the biological of this virus in detail. One of the most promising approaches for the treatment of specific viral disease is antigen technology using synthetic oligonucleotides target the region of the SARS-CoV-2 genome that across the polymerase RDRP by an RNA interference mechanism. The siRNA penetrates into the cytoplasm where it binds complementary to the target mRNA followed by request of exonuclease by the AGO protein of the melati subunit RISC complex. This results in degradation of the mRNA and blocking of translation of viral protein. The antiviral formula consists of a synthetic anti-RDRP siRNA complex with a cell-penetrating cationic peptide. This causes cytoplasmic delivery of the first formulation. Preclinical study has shown that this complex reduces SARS-CoV-2 replication in vitro by up to 10,000 times. Randomizer control open-label clinical trial results show that the drug has safety, can tolerate medicine well. That's the advantage of the siRNA method is that the molecule advances directly on the viral genome. This reduces the risk of developing.
By conclusion, it's evident that COVID-19 remains a significant global health challenge. Through sequencing of the viral genome and X-ray crystallographic analysis of the protein, research have detailed the pathogen life cycle. This understanding of viral replication, RNA transcription, and viral protein translation contribute to the development of effective COVID-19 pharmacotherapy. The advantage of antisense RNA technology is that the molecule acts directly on the virus, reducing the risk of developing resistance to treatment. Analysis of data from laboratory and clinical study indicates that current antiviral drugs can provide reliable protection against different strains of the SARS-CoV-2 virus, effectively preventing COVID-19.